which overall is just putting a bunch of rats on some drugs. The main drugs that I'm focusing on are a ADHD medication, and I'm going to talk about that in a second, as well as alcohol at a college campus. You know, alcohol is kind of common, so I'm not going to go too much into the background of that. But I am going to talk about how these two drugs interact with people with attention deficit hyperactivity disorder, also known as ADHD. So ADHD is very prevalent. About 6.4 million people just in the US alone have a diagnosis of ADHD. And so ADHD is commonly seen in children in learning environments. And students with ADHD generally have symptoms of overactivity. And so students in classrooms who have ADHD have a hard time just sitting still in one position to listen to their teacher. They also are very distractible, and so they'll look around and not be paying attention to the lesson at hand, as well as impulsive, a symptom that can cause students at younger ages to act out or say things randomly. And inattentiveness is generally another symptom of people with ADHD. And so these symptoms are treated with either medications or talk therapy. So some common medications that you've probably heard about are Adderall and Ritalin, or Concerta. Adderall and Ritalin are short-acting forms of ADHD medication. So once administered, they take a shorter time. It only lasts for so long. And then Concerta is like a longer-acting form of ADHD medication. Ritalin is actually known by Methylphenidate, that's its chemical name. So Methylphenidate is a common prescription for people who have ADHD to address all of those symptoms of inattentiveness, impulsivity, distractibility, etc. It was first formed in the 1940s and has been used to treat ADHD since the 1950s, and is also used to treat narcolepsy in some patients as well. So we're going to use a color coding system for this talk. This beautiful light blue will remind you of methylphenidate throughout the rest of the talk, hopefully. So what does methylphenidate do? It interacts with these two neurotransmitters. Norepinephrine and dopamine are two neurotransmitters that work in the central nervous system to allow different neurons in the brain to communicate with one another. So norepinephrine and dopamine are involved in centers of the brain that regulate attention, behavior, and emotion. So for norepinephrine, we're going to color that yellow, and then for dopamine, we're going to color it this purple color. So what do these neurotransmitters do? They allow neurons to interact with one another, and that's formed through an action potential. So what is an action potential? We have two neurons. This is going to be our first neuron over here, and this is our second neuron, and they're communicating with each other. So the first neuron sends an action potential down this long thing, we're calling that long thing an axon, from its cell body, that's where its center is, to dendrites on the second neuron. So those dendrites interact with the axon terminals of the first neuron and allow for a synapse to form between those dendrites and that axon. When that action potential is propagated, it looks kind of like this. So an action potential is where you have a difference in electrical potential between the inside of the cell axon on the first neuron and the outside of its cell. So here we have what looks like a membrane here, separating the inside from the outside, and we have an exchange of electrical potentials that propagates down that allows the first neuron to communicate with the second one. Once that action potential goes all the way down the axon, it'll come to the axon terminal. At the axon terminal, it propagates to allow calcium to initiate a response where little tiny bubbles that are holding those neurotransmitters like norepinephrine and dopamine to be released from the first axon of the first neuron to the synapse on the dendrite of the second neuron. After that, those little bubbles are released, and the drugs, such as norepinephrine, here in the yellow, right, interacts with specific receptors on the second neuron, causing a response of that second neuron. What that looks like in a particular scenario for methylphenidate is the idea that the first neuron releases norepinephrine or dopamine, and those two neurotransmitters interact with the second neuron. after they're done interacting with the second neuron, they'll usually be taken up by the dopamine transporter or the norepinephrine transporter, respectively. So these little boxes would go back into their homes and be taken back up by the first neuron. What happens with methylphenidate is it blocks the ability of those transporters to put the neurotransmitters back into the first neuron. So methylphenidate competitively binds to that site on the norepinephrine transporter and the dopamine transporter to prevent norepinephrine and dopamine from going back into the first neuron, which allows them to further interact with the second neuron. So what that means in terms of what we're looking at in this study is there's a lot of communication of norepinephrine and dopamine in the central nervous system, and this can be affected by multiple drugs like methylphenidate, but also like alcohol. And so alcohol is commonly taken by people with ADHD who are also on their ADHD medications. And then methylphenidate, being one of those ADHD medications, is prescribed very regularly, and this allows people to overabuse both substances. So in a recent survey, there's about a report rate of 90% of people who are on their medication while also drinking. And these interactions at those synapses between neurons changes those communications between those neurons. So how are we going to be able to observe what those action potentials look like between those neurons? We use a flash evoke potential. So flash evoke potentials is when you're using a bright light stimulus to cause an action potential, and you have a recording of an electrode in a specific area of the brain to measure what that flash does to that electrical potential under different conditions. So this is what a typical flash evoke potential looks like. There's different peaks that are labeled, right? For this particular study, we studied two places, the visual cortex and the superior colliculus, which I'm about to talk about. Those are different areas in the brain. And we put electrodes in those respective areas to analyze what these flash evoke potentials will look like under these different drug conditions. So, this is the rat brain. Up here is the top of the head, and this is where you can see the eyes of the rat, and then this is the back of the head. And so in this brain we have the visual cortex, and we put an electrode in the visual cortex to analyze what that part of the visual system would be affected by under these drugs. And then we also analyzed the superior colliculus, which is all the way down in the brain, and so we had to insert a longer electrode into that part of the brain. And here you can see that the eyes are over here. There's some information from the eyes that stays on the same side that the eye is on and travels all the way to the superior colliculus, as well as the visual cortex. And then there's some information from that same eye that goes over to the other side of the brain, to the other side of the visual cortex. So by analyzing these two systems with this electrode and this flash evoke potential mechanism, we're able to see what methylphenidate and alcohol, and in combination, have an effect on these visual areas, and therefore the effects on the conscious and unconscious and behavioral responses involved. So why are we studying these specific structures? We're going to use this color coding system again. The green we're going to use for the visual cortex, and the red for the superior colliculus. Ideally, the visual cortex allows organisms to process the main idea of what they're looking at, and then in general, the superior colliculus has different layers to it, but the upper layers that we're particularly looking at are involved in creating an appropriate behavioral response for the organism. So they're very important in terms of how someone with ADHD would be able to perceive their environment as well as respond to it appropriately. So in the visual cortex, there's three separate areas that we're looking at: the early components, the middle components, and the late components of those flash evoke potential waveforms that we're talking about earlier. And the early is more of the sensory processing that primarily takes place, and then the middle and the late are more about the behavioral and pharmacological response to different agents happening in the visual cortex. So the overall hypothesis of this experiment is to analyze whether methylphenidate will alter the effects of alcohol, here known as ethanol for the rest of this talk, alter these effects in these two specific areas in rats. And so to do that, we're hypothesizing that methylphenidate, being a stimulant of the central nervous system, would counteract the depressive effects that ethanol usually has on the central nervous system. And then the second part of our study was analyzing whether a higher dose of methylphenidate administered would have more counteractive effects. To do this, we had a long methods section here where you have to first create a little cyborg rat. So you put this rat in this apparatus and you insert electrodes into the brain. After you cut it open and drill holes, you insert these electrodes into the respective areas. Again, we were studying the visual cortex and the superior colliculus. And then these electrodes then create a plug that allows you to plug the rat into a computer. After you do that, you're able to begin testing the rat under different experiments. And so overall, we used 40 rats. 5 of them died in surgery. 10 of them were used for the higher dose, and 25 others were used for the lower dose of methylphenidate. The testing procedure consists of 4 treatments. So we're comparing everything to a saline condition of what you would normally expect the rat's flashback potentials to look like. And then we also studied ethanol by itself, methylphenidate at 2 different doses, and ethanol and methylphenidate with one being with the low dose and the other being with a high dose. So after you have this rat, it's all prepped, it's prepared to go into this testing procedure, you give it eye drops of atropine that dilate the pupils to allow those retinas to really focus on the flashback potentials. And then you do the first injection, which is either saline or ethanol in this case, and then the second injection of saline or methylphenidate. And here we have the eye drops, so you administer those. And then every injection of the drug treatment was intraperitoneally, which is a space inside the abdominal cavity, which allows the drug to be absorbed very quickly. And then you place the rat into the testing chamber. You can see this long cord starting from down here that goes to a computer in a different room. But those cords connect all the way up here and form into a plug here. This plug gets placed inside that cap of the rat's head that connects the electrodes to the recording device. After you've done that, you wait 5 or 15 minutes. So after 5 minutes, you start a recording, and then following, after 15 minutes that it's been in the chamber, you also do another recording. So there's 2 different time stops that we're analyzing the flashback potentials at. And the recordings are initiated by a counter that allows this flashing light to go off at specific time intervals, which usually takes about 3.5 minutes for 200 flashes of this light. Those 200 flashes cause individual flashback potentials in the rat's brain that take individual data points for that one flash, and then all of those flashes that are causing flashback potentials are averaged to create an average waveform that we're going to be looking at. So place that against the chamber and everything else is dark, and so the rat's only light stimulus is the flash. After you're recording flashback potentials, we also recorded body temperature and an open field behavioral analysis that I'll talk about. So 2 behavioral analysis variables that we studied were the rat's body temperature, and so we used a rectal thermometer for that, and then we also studied inside the testing chamber that the rat is in, there's light beams that go diagonally back and forth that allow you to count how many times the rat interrupts those light beams, which shows you how much the rat is moving around in the testing chamber. And then we also did an open field maze, so this is what that kind of looks like. This is an above view. You place every rat in square 16, and then every time all 4 of the paws cross over into a different box, that's counted as one movement. And so we measured the ambulation of the rat in these boxes and the differences in that given different drug treatments. We also took a measure of how many times it stood on its hind legs. For example, very drunk rats don't feel comfortable standing up. So after that, every rat was sacrificed, and this is a nice slice of a rat's brain that we're going to be looking at. And this is images that I took of all of the rat's brains to measure where that electrode went down into the brain. This is the lowest point of where that electrode met in the superior colliculus. And so analyzing that's in the superficial layers of the superior colliculus is very important because that's then recording from a section of the superior colliculus that generally has behavioral outputs. And these are images of all of the ends of the electrodes for all of the 20 rats that I did at the low dose of methylphenidate. 6 of the rats were done by another member of the lab, Sadie Tempest. So this is where all of her electrodes ended up in her superior colliculi of her rats. And then at the high dose, this is where all the electrodes ended up in the spherical nucleus of the 10 rats studied. Following that, there was also a statistical analysis to analyze the overall effects of these different variables that we studied. So, results for the first study indicate that we have this giant thing over here. These are the waveforms that I was talking about, right? They're summations of all of the flashback potentials generated by 200 bright flashes while the rat is in the testing chamber. And so up here we have saline, and every peak is labeled according to how long it took for that peak to occur based on when the flash was initiated. So these long dark lines are when the flashes were initiated, which is about 50 milliseconds after the beginning of the recording took place. And so that allows us to label each peak a little bit differently. But for the purposes of this talk, we're just going to label them P1, which is a positive component, N1, which is a negative component, P2, N2, P3, N3, and P4. So that's in the visual cortex. We studied these 7 different peaks. And this is a way that we study them, which is through amplitudes and latencies. So amplitudes indicates how big the peak is in comparison to the baseline, and then latencies indicates how long it took for that peak to occur. And so we analyze the differences between amplitudes and latencies for each of the peaks under each of the conditions. And the interesting results is what I'm going to share, not all the results because that would take a while. So for example, in the amplitude one that we're sitting right here, this is that peak, and that peak is right around here, so that's this big broad one. And so we took the top of that peak for each treatment. And the key things that we found was that the effect of ethanol was statistically significant. Ethanol for all of these graphs is seen in red, and then methylphenidate is seen in blue, and when you add blue and red together you get purple, so the combination treatment is in purple, and then the saline is in black. So ethanol here in red showed an overall significant effect as compared to the saline condition. Methylphenidate, which is the blue up here, showed an overall significant effect also compared to the saline condition, which is right here. And then there was also a significant interaction between the two, which is the purple line just above the saline. And so the overall significant effect could be in comparison to the saline, but also in comparison to the ethanol or the methylphenidate. For the latency, for example, we're looking at this component, which is this broader one down here. This is a middle component, so that's more associated with behavioral outputs in the visual cortex. And we analyzed the control is black here in the middle, and found that time was a significant factor. As you can see, the latency was generally a little bit longer for this component at the first timestamp, but shorter at the second timestamp. And we also found that methylphenidate by time had an interesting interaction, which is this blue line, and then methylphenidate, ethanol, and by time also had an interesting interaction where, contrary to the rest of the groups, there was actually an increase in latency. So what that would look like is this component right here is longer than the rest of them. For the higher dose of methylphenidate, we found other interesting results. So generally for this component, which is that one that we were just looking at before in the first study with low methylphenidate, had very interesting results where all of the variables were affected by the drug treatments and by the effect of time. Amplitudes generally decreased for this particular peak compared to the saline, which is very interesting, and then time also had a significant effect of decreasing the peak as well. For the latency, we're still looking at this first peak here. So that's that first P1 that we're talking about right after the flash evoked stimulus. This is more involved with sensory processing. And so for this particular peak, there was significant effects for ethanol, methylphenidate, ethanol by time, methylphenidate by time, and the ethanol methylphenidate by time. So we can see that the saline condition down here had a decrease in the time it took for it to appear in the second time stop, but ethanol severely increased the time that it took for that component to appear. And then the combination treatment, which I find the most interesting, actually had even more increasing effects compared to the drugs alone. In the superior colliculus, at the lower dose of methylphenidate that we studied, there was a significant effect of ethanol on this one component that we're looking at here. For the superior colliculus, we're only studying 3 different components, P1, P3, and N4. And so by studying these components, they're generally involved in behavioral outputs. And so they're more specific to the general scope of this type of study. But for this particular component, that would be P3 over here. This one has significant effects of ethanol that decrease the amplitude of that component. As you can see here, it is much less negative than it is in this one. So methylphenidate actually had an increase in the amplitude, as you can see here. And the interesting part is that methylphenidate counteracted the effects of ethanol by bringing it back more to the baseline saline conditions in the combination treatment. For latency, there was an interesting effect of ethanol only for this same component that we were just looking at. So it took about the same time for that peak to appear for the methylphenidate and for the ethanol and methylphenidate combination. But there was a significant difference where you can see ethanol here severely increase the time it took for that peak to appear in the superior colliculus compared to the saline conditions. Now you would think that the combination treatment seen right underneath the ethanol treatment had a significant effect, but it wasn't statistically significant, unfortunately. Moving on to the second study, at the higher dose of methylphenidate, we're studying the last component here, which is N4. N4, as you can see up here, allows you to see that there is a significant change in the ethanol as well as the methylphenidate, which severely decreased the amplitude of that component. And the combination brought it closer to the saline, but it wasn't statistically significantly different. And then for the latency, you can see that this combination treatment actually was summarization of both the ethanol and the methylphenidate effects of increasing the time it took for that component to appear as compared to saline. And so N4 took a much longer time to appear in the combination drug treatment. For our behavioral analyses, we studied 4 different things: the ambulation of the rat in the testing chamber, the ambulation of the rat in the behavioral maze, the ability of the rat to stand up in the behavioral maze, and body temperature. The most interesting one is the one highlighted in purple here, which is the effects of the combination drug treatment of methylphenidate and ethanol to severely decrease body temperature, inducing severe hypothermia, and this is just at the low dose. Other key findings also show that there was an increase in ambulation movement for rats inside the testing chamber under the combination drug treatment as well. And the difference here is that there was not a significant increase in the number of times the rat stood up when it was underneath the drug combination treatment as well. For the second study with a higher dose, that actually completely changes, where you can see that there is a severe increase in the number of rearings that occurred for a rat when it had methylphenidate at the higher dose of 11.6 milligrams per kilogram of rat compared to the lower dose before, when ethanol is all the way down here. So that was very interesting. And then there was even more severe hypothermia produced by the higher dose of methylphenidate as well. So the main results of all of this information that I just threw at you is that the low dose of methylphenidate produced some significant results on the Flashback Potentials and the behavior and the body temperature that were replicated again at the higher dose in more severe notions, but the higher dose was able to counteract the effects of ethanol in specific situations, such as the superior colliculus. That was more statistically significant, where methylphenidate counteracted the depressive effects of ethanol in that brain area. And then lastly, hypothermia was a big thing, and so analyzing that both drugs alone, ADHD medication and alcohol, have severe hypothermic effects, but in combination cause a rat to be very, very cold. So following that, the idea that methylphenidate had an increase on ambulation compared to the severe decrease that ethanol causes a rat to not want to move around, it was able to do that only at the high dose and not really at the low dose. Lastly, the idea that methylphenidate does not conclusively counteract the depressive effects of ethanol are the main findings of this study, and we hope to be able to analyze that in further doses in the future. The core take home is taking Ritalin while drinking is a very hazardous idea. Don't do it. You're going to end up like this guy otherwise. So I would like to thank all of my contributors, my primary mentor, Dr. McLester, his amazing wife, Leroy from the physics department, Dr. Martin, Daniel Martin, for providing some great ethanol. That was great. Previous lab members, including Sadie and Josephine, who really helped for the overall conclusion of the study, and then also all my friends. Thank you for coming, and my family. Questions?
There you go.
So, can you talk a little bit about the I know this might not be interesting for you, but I'm really curious about the process of when you actually have to cut open the rat's head and how, what is that process like when you're actually inserting the probe to a certain region of the brain? Number one, I guess, is do you have to is there any sort of pain relief you have to give the rat? And then also, how do you, I know you measured the depth of the insertion, but when you're actually doing it, how do you, that seems like you have to be really careful because it's so small, how do you actually make sure?
Yeah, the whole surgery site is probably about 10 millimeters, and so it's really tiny, and the process of using a stereotaxic apparatus is what really allows you to do it. So you prepare the rat by knocking it out with pentobarbital was the solution that we use. There's a few others that you can use to do stereotaxic surgery, but that's the one that this lab does. And so you knock it out, and then you shave its head, and then you put lidocaine underneath the surface of the skin so that there's no pain there when it wakes up. And then you scrape away the membranes across the skull and clear off any blood, and then you measure the specific holes that you're trying to drill in. So here you can kind of see that there's multiple holes, and there's reference points on the brain skull, primarily bregma. It allows you to measure from those specific distance points that are the same in pretty much most rat skulls. That's been a tried method before. And so measuring from those reference points, you can idealize where you want to put your electrodes. And then measurement of how far down it goes is actually by this awesome stereotaxic mechanism that has specific millimeters, and so you can say that the superior clavus is 4.7 millimeters down from the skull. Yes?
So in the neurotransmitter picture that you had with the, thank you by the way for the color coding, that's so helpful. So you had the pink and the purple, no, sorry, not pink and purple, yellow and that one, thank you. So the MPD by itself,
Yes.
and then with the addition of the ethanol, can you clarify for me how exactly do your results at the end that showed that the ethanol and the MPD combined caused more of an effect, how would that work here?
Sure, I should have clarified that.
No, that's my own, I'm not familiar with your own things. So.
Ethanol has multiple effects on many different neurotransmitter sites, one of them being dopamine. And so the idea that ethanol affects the dopaminergic actions that are involved in the visual system is the primary background to this research. And so by methylphenidate affecting that, also the idea that it would be more of a competitive inhibitor compared to ethanol and allow for more dopamine to be released instead of ethanol binding to those dopaminergic sites on the second neuron.
Okay, so there's a yellow and purple on the second neuron as well. Okay. or?
Yes.
Okay.
So these neurotransmitters, norepinephrine and dopamine, are released by the first neuron, and then they go and interact with the second neuron, which is where ethanol would normally interact. And then to allow them to interact more, methylphenidate blocks their ability to be taken back up into the first neuron.
Are there other, so you did Ritalin and Adderall, are they similar enough in chemical composition that these results would translate to the other drug, or is this pretty much specific to?
Yeah, that's a really great question. I know that there's been a lot of research. I would have to look this up for you, but I know there's been a lot of research in terms of the bioselectivity of methylphenidate compared to amphetamines, et cetera, cocaines. But I'm not entirely sure if particularly a cross study of how effective on these specific sites that different types of ADHD medications have been. But Ritalin is like the short-acting form of methylphenidate, and then Concerta is like the long-acting form. Adderall is a little bit different, but yeah.
Follow-up question. So can you talk a little bit, so we know that methylphenidate and ethanol are both going to be processed by enzymes in the body called cytochrome P450s. So I would ask, so you gave a very nice explanation to Dr. Fleshman, but I'm going to wonder, is there competition between the methylphenidate and the ethanol because they use the same cytochrome P450, or do they use different ones?
That is a great question that I don't know the answer to, but I can look it up for you. Any other questions?
Oh, come on, you guys. You knew I was going to ask that. Other questions? All right, well, let's thank the speaker. So you ready to lose in darts? Wow, that's just...
Yeah.
A little extra by the third.
Yeah, so normally, Parkinson's prescribe all dopamine to reduce more dopamine. So the neurons that we normally be getting a lot of dopamine aren't. So similarly, like ethanol is blocking the ability to hormone of dopamine to interact.
And I just sent you the link to YouTube videos. There we go.
I watched it.
There you are.
Is there any chance that you could take out the first 3 minutes?
Thank you.
Also, oh, I am sorry. Yes, I can.
I think it probably wasn't the first thing.
It was for the, and I don't have access to the other link, so I will email the guy and ask him to move it. Yeah, oh, did I turn that on? Oh, no.